along scenic coastlines in the south. Waves of mesmerizing green and golden grass stretch to the horizon. This is the salt marsh. Part liquid, part solid. An ever-shifting landscape, stretching for miles before it meets the sea.
Coastal marshes are found anywhere there's soft sediment and not too much wave action. In the US, our big marshes are in South Carolina and Georgia, and then areas like Louisiana.
There's a huge amount of life that lives here.
Marshes function as nursery grounds for a number of commercially and recreationally important species. Everything from shrimp to crab to fish that are very popular with sportsmen.
Between the ebb and flow of the tides, the only constant is change.
Fish will move onto the marsh at high tide to feed.
And at low tide, you'll get more terrestrial organisms.
Herons and egrets and ibis, spoonbills, all of them feed in the marsh.
And organisms that are in the mud that come out, like the crabs. So it's really fluid.
Marshes filter nutrients and contaminants from the water and serve as buffers during storms.
They provide protection from wave energy, from hurricanes.
It is an absolutely beautiful landscape.
I mean, I love everything, including the smell of a healthy salt marsh on a moist morning.
They really give a sense of place to Georgians, South Carolinians, North Carolinians, and Floridians.
As we're worried about climate change, marshes are one of the important habitats that can help take up carbon and store it rather than have it be in the atmosphere.
How do scientists study the salt marsh? And how resilient is it to climate change? The southeast coast of the United States is famous for its extensive salt marshes. In coastal Georgia, moss-draped live oaks give way to vast expanses of grass, broken up only by winding creeks.
We have a very beautiful wild coastline.
I think the people in this state really appreciate the marshes, and it is part of the identity.
We have about 350,000 acres of salt marsh here on the coast of Georgia, which is about a third of what you have on the East Coast today.
We only have 110 miles of coast, but I heard once that if we measured all the tidal creeks, we have about 5,000 miles of shoreline in this marsh habitat. Georgia has several factors that give us so much marshland, and one of them is the tidal range, and that is due to the shape of the coastline and the gradual slope of the continental shelf.
Our average tide is about 7 feet, and it can be higher during some parts of the year, so that's a pretty big shift from high tide to low tide.
Most of Georgia's 14 barrier islands remain largely undeveloped and wild, including Sapelo Island, about an hour south of Savannah. It's home to the University of Georgia's Marine Institute.
There's a long history of salt marsh research here at the University of Georgia Marine Institute, and it dates back to the middle of the last century when a number of famous ecologists started working on the Georgia salt marshes. And so there's just an enormous amount of knowledge that's been gained over the years that really makes this location the premier place in the world to study salt marshes.
We really are standing on the shoulders of giants.
And the cutting-edge research continues. Dr. Merrill Alber leads the Georgia Coastal Ecosystems Long-Term Ecological Research Project, which currently involves 22 scientists from 10 different universities.
We've been studying the marsh here since 2000. The overall underlying theme is, from an ecological perspective, understanding long-term change.
One key component of the project is the annual fall monitoring.
That shows us what's happening over decadal scales in the system.
We do that in the fall because that's the end of the growing season.
We have a network of 10 sites, and so the idea is to assess the functioning of the marsh from year to year. And we've been doing that now for 21 years, and we're starting to see interesting patterns of what affects the plant productivity in particular.
Along the southeast coast and into the Gulf of Mexico, Spartina alterniflora, commonly known as smooth cordgrass, is the dominant grass species found in tidal salt marshes.
The biggest thing that drives how vigorous the plants are is how much fresh water is coming down the rivers. And what that fresh water does is it makes the marshes a little less salty. Even though these are salt marsh plants, they still have to deal with salt being a physical stress to them. And so a little less salt means they can grow a little bit better. And we see 2 to 3 fold differences from one year to the next in how much biomass there is at the end of the year. Another thing that affects the productivity of the marsh plants is temperature. So in hot years, the plants don't grow as well. And that, of course, is a concern when we think about climate change.
The scientists also count the number of invertebrates found in the marsh.
There's the periwinkle snail, and that feeds on fungus that is decomposing the marsh plants. In doing so, if there's a lot of snails, they'll damage the plants as well. And so when the snails are really abundant, they can suppress the biomass in the plants. A second common invertebrate is fiddler crabs, and they are what we would call an engineer because they dig burrows which improve the nutrients getting into the soil. And so they stimulate plant growth. The third invertebrate is the marsh mussel, and they filter the water and deposit sediments and waste products onto the marsh surface. And so they're building marsh elevation and also fertilizing the plants. Here's the next one right here.
Starting in the late 1950s, salt marsh ecologist Dr. John Teal was the first to study the productivity of the marshes surrounding Sapelo Island. He wanted to know where all the carbon entering the marsh ends up. Carbon is one of the key building blocks of life, which cycles through the living and non-living parts of the environment.
We can think about how much the plants grow and how much energy is going into that plant growth, and then how much production went into the various organisms that might use it in the marsh, how much of it goes into the atmosphere, how much of it goes out with the water and then fuels the coastal ocean.
More than 60 years later, scientists are still investigating the answers to these same questions. Since 2014, high-tech instruments are helping to track the movement of carbon in the salt marsh.
This is the Flux Tower, and we actually are collecting every 5 minutes 88 different environmental variables from 21 different instruments. And in addition to that, with the carbon flux itself, we're collecting 16 variables at a much higher frequency of 10 times a second, which adds up to 864,000 measurements a day of just CO2 flux data.
In particular, we're looking at carbon dioxide, so we're looking at carbon exchange between the marsh and the atmosphere.
Research has shown that coastal wetlands, such as salt marshes, mangroves, and seagrass beds, can bury carbon at a rate that's 10 to 100 times higher than upland forests.
And that's a very active area of research, trying to understand how much carbon can we bury, how long will it be buried for, and is this a way that we can try to decrease CO2 levels in the atmosphere, which are leading to warming trends right now.
To better track the fate of the carbon, the researchers are looking at the above and below ground biomass of the plants by taking monthly cores of short, medium, and tall Spartina found in different areas of the marsh.
The plants grow taller as we get down closer to the water.
Once back at the lab, the scientists wash the mud off the plants. Different sections of the organic material are then put in a drying oven and weighed to determine the biomass contained in each sample.
We're very interested in the differences between the forms of Spartina, the tall versus medium versus short, because there are differences in the amount of below ground material. Even though the tall plants are very tall on the surface, there's actually less below ground material associated with them than the shorter forms. So we have a habitat map of the whole area, and we know where the tall plants are found in the medium and the short, so we can extrapolate that to get information on the biomass patterns through the whole system. And then we can use remote sensing and satellite work, and we're able to then look at the change in below ground biomass based on the above ground characteristics.
We've seen some evidence that we're losing some of that below ground, which is worrisome. We're concerned what that might mean for the plants to thrive, but also for the plant's ability to hold that sediment in place.
The scientists think the loss of below-ground biomass might be caused by an increase in sea levels. How well salt marshes fare as sea levels rise is partly determined by how much sediment is being built up on the marsh. Until recently, Georgia's marshes were thought to be relatively robust, especially compared to the marshes elsewhere in the United States.
From the work that we've done over the last 20 years here along the Georgia coast, we find that marsh accretion rates are somewhere between 1 millimeter to perhaps 3 millimeters per year. The average rate of sea level rise over the last 80 years is about 3.5 millimeters per year. And so in general, we find that the marshes are not keeping up with the rate of sea level rise. Luckily, our marshes have something called elevation capital, which means that they sit a little bit higher in the tidal frame, and so they have a ways to go before they start drowning. But at current rates of sea level rise, it doesn't appear that the marshes are keeping up.
The models that predict sea level rise say that by the year 2100, we will start to then see a rapid loss in the Georgia salt marshes.
The future impacts of sea level rise not only pose a threat to the marsh itself, but also to the humans living on its edge. This is particularly true on barrier islands, like the popular tourist destination, Tybee Island, near Savannah.
Living on a barrier island, you have the ocean on one side and you have the extensive back barrier environment, which is typically filled with salt marsh, on the back side. On the front side, we know how to protect ourselves from storms, and we know that we need to build large sets of sand dunes, which serve as sacrificial erosional features that protect our upland infrastructure. On the back side of the island, the answer to storm flooding is not quite so clear. The salt marsh extends right up to the upland, and there are many houses that are built near the edge of the marsh, so it is not obvious how you would build some kind of feature to stop back barrier flooding.
Sedimentary geologist Dr. Clark Alexander, together with other scientists from the University of Georgia, is working with the city of Tybee to figure out potential solutions to protect the backside of the island from flooding in the future.
The city of Tybee Island has been very proactive. They retrofitted a number of their storm drain systems, so when storm tides were high, water didn't come up the storm drain system and flood the uplands They've looked at building a wall around the backside of the island, but it's actually not financially possible.
Tighten up that nut as tight as you can.
To determine how much sediment is accumulating in the marsh behind the island, scientists take a 1.5 foot long section of the mud.
And then we're going to take it back to the lab. We're going to slice it up into thin sections vertically.
By looking at the half-lives of naturally occurring radionuclides in those sections, the scientists can determine how much and at what rate sediment has accumulated over the last 100 years or so.
And that will give us some idea of how these marshes are keeping up with current rates of sea level rise.
Salt marshes also occur along the northern portions of Florida's Gulf Coast. Scientists with the state's Fish and Wildlife Research Institute's Garson Point Lab near Pensacola study a variety of fishes that spend time in the marsh. The tiniest is the salt marsh top minnow, which is thought to be rare and declining in numbers.
Salt marsh top minnows inhabit tidal marshes along the Gulf of Mexico from Texas to Florida. And in Florida, they only occur in the northwestern portion of the state.
Chelsea Miles McBurney has spent the last 5 years monitoring 32 randomly selected sites in the marsh to figure out where these fish live, what sort of habitat they prefer, and if their population is stable. The salt marsh top minnow was listed as threatened by the state of Florida in 1977 due to its restricted range, diminishing habitat, and population fragmentation.
The species is actually currently considered a federal candidate for listing by the U.S. Fish and Wildlife Service.
To catch the tiny minnows, experts use 2 different types of traps.
And we set those in a 12 by 16 meter grid alternating trap type. So we set those in the afternoon, and we'll pick them up in the morning, and all fish that are in the trap get identified, tallied, and released. And for all salt marsh top minnows that we come in contact with, we will take the total length.
All right, he is 3.5 centimeters.
Record the reproduction status. We also record a lot of habitat parameters, including the type of vegetation around the trap.
Percent veg, 5% Spartina.
Salt marsh top minnows prefer typically low to moderate salinity levels, so that true brackish environment. And they can typically be found along the edges of marshes or within those intertidal creeks that are within the marsh.
Experts also take small fin clips for DNA analysis. This research has shown there are 5 genetically distinct groups of salt marsh top minnows in Pensacola Bay. And while the overall population appears to be stable, future research is needed to identify specific threats to each of these subgroups and the potential risk of extinction. Another fish that spends time in some of Florida's salt marshes is the rather large and prehistoric-looking alligator gar.
Primarily they're freshwater fish species, but we're finding out they have fairly high tolerances for salinity, and so they can move in between fresh and brackish and even full-strength saltwater fairly easily. Alligator gar occur west of the Apalachicola River here in Florida, and that extends on into Texas and down south into Mexico, and then as far north as Illinois.
In 2006, the Florida Fish and Wildlife Conservation Commission closed the harvest of alligator gar in state waters.
There was some concern that alligator gar populations in Florida were going down, and they wanted to step in and do something before it was too late.
Since 2010, the Garson Point Lab scientists have been conducting research in Pensacola Bay and its tributaries to get an idea of how many alligator gar are found there.
Around 2015, we were able to estimate there were about 212 alligator gar in the Escambia River. From there, we decided to take what we had learned on the Escambia River and apply it to other rivers.
To catch the fish, the scientists set large mesh gill nets. Once an alligator gar is caught, the experts collect a variety of data.
186.
Alligator gar are able to breathe air and are fine out of the water as long as they're kept moist.
There are several species of gar here in Florida, and at first glance, they all look similar. But there are some major differences between them, namely the size of the fish.
Alligator gar can weigh over 300 pounds and grow up to 8 feet in length.
Back in the 50s and the 60s and even into the 70s, they were eradicated because they thought they consumed sport fish. And we're finding out that's not the case.
To track the apex predator's movements, the scientists implant an acoustic tag into the fish. This tag gives off unique pings that can be picked up by more than 70 acoustic receivers located throughout the bay.
And that's like a computer that will pick up a tagged fish that swims by it. Those things are picking up information 24 hours a day, 365 days a year. The fish that we have tagged now in Pensacola Bay spend anywhere from 6 to 8 months in the brackish water habitats, and then those other 3 to 4 months in the freshwater habitats during the spring.
Once the scientists have enough alligator gar tagged from across Pensacola Bay, they plan to do a population estimate for the entire watershed and determine which river the animals use to spawn. The acoustic receivers scattered throughout Pensacola Bay are also used by Garson Point Lab scientists studying Gulf sturgeon.
They have changed very little over the span of millions of years. Essentially, they are a swimming dinosaur. Gulf sturgeon range eastward from the Suwannee River in Florida over to the Pearl River in Louisiana.
Once coveted for their meat and caviar, the Gulf sturgeon population has declined dramatically and has been listed as federally threatened since 1991.
What we're not seeing is an expansion, as you would expect, 35 years post-listing, and so that's what actually has guided our research. Adult sturgeon appear to be fairly stable. We think that there's some sort of population bottleneck in the early life history stages of sturgeon. What we're referring to is generally the 0, 1, and 2 age group.
708.
The scientists are part of a larger research team involving multiple partners, studying juvenile Gulf sturgeon across their range. In the spring, the fish move into freshwater to spawn. They remain there until late fall.
And they may fast for anywhere from 6 to 8 months in these freshwater habitats.
Tagging data has shown where the sturgeon go once they leave freshwater.
Essentially, the adult sturgeon will outmigrate very quickly, and they will move beyond the salt marshes to either sound habitats or even offshore habitats. What we've observed with juveniles, specifically in the age 1, 2, and 3 range, is that they have an affinity for low salinity habitats just outside the river mouth. They exclusively occupy salt marsh habitats.
Researchers say they hope to learn more about the survival rates of the juvenile fish.
And if survival for these early cohorts is particularly low, then that allows us to sort of hone in from a management perspective on we need to really determine what's going on in these estuarine and these salt marsh habitats, because that appears to be one of the limiting factors to recovery of the population.
America's southern salt marshes are highly productive ecosystems, linking the land and the sea. And they're also a place of timeless beauty.
It's a real wilderness.
Just being in this beautiful, peaceful place, I think is good for the soul. It's hard to put a value on it.
DSV Alvin, also known as Deep Submergence Vehicle 2, is a deep-sea submersible owned by the United States Navy and operated by the Woods Hole Oceanographic Institution.
Stop, seal the hatch.
Carrying 2 passengers and a pilot on each dive, the sub has taken thousands of explorers to depths once thought unreachable.
Every time I get to dive in Alvin, I see something I've never seen before. I get to experience that environment firsthand, and it's often led to big discoveries, not only for my research, but other people as well.
For more than 50 years, the sub has traveled to extraordinary places, capturing the world's imagination and heartbreak. In 1986, it dove on the Titanic, deploying the remotely operated vehicle, Jason Jr., and returning with striking imagery of the site. And following the Deepwater Horizon disaster in 2010, Alvin aided scientists investigating the impacts of the oil spill on deep-sea corals.
What sets Alvin apart is its productivity. It's incredibly well supported by the community of researchers that use the vehicle and the National Science Foundation and the Office of Naval Research.
Of its thousands of dives since 1964, there is one often held as the most significant: the discovery of hydrothermal vents near the Galapagos Islands. These alien-like deep-sea communities, where life thrives without the light of the sun, were unknown to humankind before Alvin visited them.
The discovery of vents changed the way we think about life, and Alvin was really the workhorse that did all that work in 1977.
If you look at it on a map, it just looks like a big blue expanse, but down on the bottom, it's a different planet.
Now, Alvin is poised to continue its revolutionary scientific work. With a new set of upgrades and deeper depth rating, the possibilities are endless.
Alvin will be able to map things that we've never seen before, that otherwise have not been accessible to human discovery.
How has the research conducted with the Alvin submersible impacted our understanding of the oceans? And what can Alvin's incredible history of discovery, inspiration, and daring tell us about life on Earth and beyond? At the National Deep Submergence Facility at the Woods Hole Oceanographic Institution, or WHOI for short, a team of engineers is working on improvements to the human-occupied vehicle, Alvin. This upgrade will take Alvin's cruise deeper and farther than ever before.
Alvin undergoes a complete overhaul every 5 years or so. The sub is completely taken apart, everything is serviced, and then it's put back together.
This $8 million upgrade will expand the submersible's depth rating from 4,500 meters down to 6,500 meters, roughly 4 miles below the ocean surface. Soon, scientists will be able to reach nearly 98% of the ocean in Alvin.
current update to Alvin that's happening right now is going to be fantastic for us. We're having new high-powered thrusters. We're having new motor controllers, all the things that help us be more agile on the seafloor, things that allow us to get down to deeper depths quicker. The control systems on the vehicle are going to be fantastic, the new upgrade of cameras and lights. Alvin has been the workhorse, and it's always evolved with the science that we want to do. There's virtually no part of Alvin today that is original to Alvin.
The Alvin submersible has been updated so many times since 1964. It has been likened to the ship of Theseus, a thought puzzle over which philosophers such as Plato, Plutarch, and Locke have deliberated. Is a ship still the same vessel if it has been entirely replaced over time, piece by piece?
It's a story of a ship that perpetually exists, but all the bits and pieces have been replaced over time. We always get this question, what's the oldest part of the submarine? And it has evolved over the years, right? Alvin is not the Alvin that it was in the 60s by any stretch. And it's not the same sub that was in the 80s. When we come out of this overhaul, it's likely to be no later than about 2012. So it's same, but different, right? The technology that's been brought on board the sub has enabled it to last easily another 50 years. So it's a platform that I really hope sticks around and enables things that we can't even think of right now.
More than 3,000 people have explored the ocean floor in Alvin on over 5,000 dives around the world.
I have used Alvin for a number of my deep sea studies, working in the Atlantic and the Gulf of Mexico and the Pacific Ocean. I've had really incredible success using Alvin to conduct very complicated science in extreme deep sea environments. And we use Alvin to explore, but also do really systematic ecosystem-based science in these different places.
Researchers from disciplines across the sciences have used Alvin to study the ocean's depths, from biologists and chemists to geologists and volcanologists like Patricia Gregg, who studies volcanoes on Earth and other planets.
Over the past few years, we've been looking at a series of volcanoes in the equatorial Pacific. And one of the things that we were very interested in with this chain of seamounts was whether or not they could be potential oases for marine life moving up and down mid-ocean ridge axes. So is this something that could allow biology to make its way along the seafloor? So understanding these volcanoes and their eruption history could give us a lot of information for how life has evolved and moved in our water columns.
Tomorrow morning it starts. My first Alvin dive. Very excited, a little nervous, just a little nervous. Okay, a lot nervous.
One of the things that I did on my first dives was I videoed myself.
Just waiting to go.
Diving in Alvin for the first time is often a transformative experience, especially for early career scientists, sometimes changing the course of their studies.
Dive 2683, December 13th, 1993. You never forget your first dive. I was blown away, and this is what I was going to do for the rest of my life.
I was so excited about diving in Alvin for the first time, so I think I woke up not really having slept and being incredibly thrilled to have the opportunity to go inside.
When you're going down, to conserve energy, the pilots turn off the lights. So it's completely black except for a few blinking lights. And then just before you get to the bottom, the pilot flips on the lights.
Everything was completely still, so it looked as though you could just open the hatch, get out, and walk around outside. Of course, you can't do that because the pressure at that depth is around 2 tons per square inch. So it just blew my mind.
You know, the bottom of the ocean, if you go down more than 1,000 feet or so, it's about like a refrigerator. So it's chilly. You're looking at hydrothermal vents, so like, you know, literally a few feet in front of your face is something that's venting 400 degrees Celsius, which is like, you know, 750 degrees Fahrenheit. And it's just amazing and mesmerizing.
Time just flies by. It goes so fast. There's all of this work and anticipation and effort and logistical challenges to just get the vehicle in the water. And then once you're there, you're on a clock. You're going down and actually representing the science of all the people that are involved in the research, not just your own work. So you're expected to do a lot of different tasks in a minimum amount of time.
Alvin goes in the water at 8 a.m. and it comes up about 5 p.m. So it's a full day in the water. I think the most surreal aspect of it is eating lunch. And you're just sitting there eating food, and you kind of take a break for a minute so it can all sink in while you're eating, and you're like, there's like 2,500 meters of water on top of me, and that's ridiculous. I feel like I spend the whole dive either with my face up against the window, just staring out the window in amazement, or vigorously taking notes because you don't want to miss anything.
I get to see this experience on a regular basis when I take people in there. They're transformed. Their ability to understand the planet has been altered, and it all goes back to the machine and the vision of the people who created it, right?
This is Alvin. It opens a new frontier of research. Now man can explore the ocean to the very outer edge of the continental shelf and beyond.
If you could go back to those original days and let them know you will be creating something magnificent, it'll have such a huge impact on people's lives, much less the greater scientific community. Maybe a few people had a vision of that, but the scope of what they created is pretty magnificent, and it's lasted.
Not long after Swiss scientist Auguste Piccard launched the first deep submergence vehicle in 1953, the bathyscaphe Trieste, Woods Hole scientist Alvin proposed the United States develop its own.
I had been one of those people who felt the submersible had a future. It seemed logical that rather than building another general purpose research ship, it would make more sense to build some special purpose ships that could go down to the bottom and look with the eyeball, so we could look over a terrain that was otherwise not accessible. And in actuality, the geologist has long been associated with the burro or the mule, and we wanted some kind of a mule that would dive.
By 1962, Huey had partnered with the Office of Naval Research and the Aeronautical Research Division at General Mills to build the world's second deep submergence vehicle. The new submersible, capable of diving to 6,000 feet, was originally nicknamed Sea-Pup, but the newly formed Huey Deep Submergence Group instead began calling it Alvin, after Alan Vine, who had championed its creation.
They probably didn't realize at the time that in calling it Alvin, they gave it life. If you ask me, it's a living thing. It has a personality, there's no doubt in my mind. So certainly people think of Alvin as a machine, but they really do think of it, I think, we all do, as having an existence that's beyond just this thing that's parked there on deck.
Alvin was commissioned on June 5, 1964, and its first pilot took it on a test dive in Woods Hole Harbor 3 weeks later. The team then completed 77 tethered dives to maximum depths of 70 feet, and by August 4th, they took Alvin on its first free, non-tethered dive to 35 feet.
A year later, a craftsman from Cape Cod took 2 old Navy pontoons and made a tender for it. Alvin had to have a ship, and that mothership was named Lulu, and that was after Alvin's mother, who was named Lulu.
In March of 1965, Lulu and Alvin set sail on their first voyage to Andros Island in the Bahamas.
Lulu Alvin, passing 5800 feet, approaching the bottom. I will call you back.
The next year, the U.S. Navy called upon Alvin to find and recover a hydrogen bomb off the coast of Spain that fell into the Mediterranean Sea following a mid-air collision. Alvin located the missing munition near Palomares in 2,500 feet of water. It wasn't until the early 1970s, just a few years after astronauts landed on the moon, that Alvin was used for science.
As Alvin was being born, so was plate tectonic theory. And that theory is that oceanic plates pull apart. Interior heated magma comes up and spews out onto the seafloor. It freezes quickly onto the seafloor. When it does that, it contracts, it cracks. That enables an egress for hydrothermal vent water to come through. And so the idea that scientists had, including Bob Ballard and others, was that there should be heat coming out of the Earth's crust.
French and American geologists, including Dr. Robert Ballard, began developing a multi-submersible expedition to explore the mid-Atlantic ridge. This French-American mid-ocean undersea study, or Project Famous, would lay the groundwork for what would become one of Alvin's greatest discoveries. To prepare for the mission, engineers replaced Alvin's steel personnel sphere with a titanium one, extending Alvin's depth range from 6,000 to 12,000 feet.
At that stage, we didn't know what these mid-ocean ridges really were. We knew that the continents drifted apart. We knew there was this mountain range in the middle, but what happened there, we didn't really know until we were able to get down there and validate the hypotheses that people had come up with.
The 1974 Famous expedition collected 100,000 photos and 3,000 pounds of rock samples. Alvin made 17 dives and spent more than 80 hours on the seafloor. They discovered vast fields of seafloor lava, but found no evidence of active heat vents. 3 years later, forming a new expedition, they sailed instead for the Galapagos Rift, where they hoped to more easily measure the heat in the sediment. The Galapagos Hydrothermal Expedition launched in February of 1977, using Alvin and a towed vehicle named Angus.
You tow it behind the ship, and it has a camera system on it, and it has sensors to detect temperature. And so they would lower this over the seafloor and drive it around. They would take 3,000 images at a time. They'd bring that vehicle up. They would develop the film on board the ship and look at the roll of film. They saw nothing but lava for thousands of images, and then they had 13 that showed these massive clams that looked to be alive. And then after that 13 images, it went back to lava again. And so they dove Alvin, dived 713, and they found one of the greatest discoveries, I think, in the history of deep ocean or marine science. They found superheated water coming out of the seafloor, which they were looking for, but they didn't expect to find the mass amount of animal life that's there.
Imagine if you were sitting in this tiny little vessel, looking out the window, and you were the first people to actually observe giant tube worms in their natural habitat, right in the middle of a volcano on the bottom of the ocean. So they were literally experiencing something that fundamentally changed the scope of what we know about the planet, but also in that moment for them, what a great experience. I think they had a good idea of what they were going to see, but I'm sure when they actually saw it, it blew their minds.
The discovery of hydrothermal vents opened an entirely new field of ocean science and led to discoveries of vent systems around the globe, including the highest temperature vents, called black smokers, where iron and sulfur combine to create black chimneys along deep cracks in the ocean's crust. Ocean scientists have built upon the initial finding ever since.
In 1991, a group of scientists, chemists and geologists and biologists were going to the East Pacific Rise to go look at vents that had been discovered 2 years before. And they dove with Alvin to where they saw hydrothermal vents, chimneys, lush communities of life there, and all that was gone. Lava had come out through fissures in the seafloor, come out, wiped out all the communities, gone drained back into the earth, and where it cooled and froze instantaneously on the seafloor, cracks formed. And so new venting came through those cracks. So it was my PhD project to look how the animals came back and recolonized these new vents.
Dawn Wright and Tim Shank were still graduate students when the Alvin expedition, dubbed Adventure, serendipitously happened upon this recent eruption in an area on the East Pacific Rise known as 9 North, and witnessed what became known as the tube worm barbecue.
1991 was the closest that any scientists had come to actually witnessing a volcanic eruption on the seafloor in real time. It was determined that their dive was within 2 weeks of the actual volcanic eruption. So we were all blown away by that. Because of that discovery, that particular area was really what they called ground zero. Subsequently, there were literally hundreds of discoveries of new species. So one discovery certainly leads to another. And now we can keep going back to the area, sampling, observing, mapping, to see how a hydrothermal and a volcanic system evolves from that time zero.
The work to understand these otherworldly ecosystems continues today as scientists use Alvin to explore and build upon the initial hydrothermal vent discovery nearly 45 years ago.
Most of the work that I do and like all of my colleagues is because of that one discovery. That was in 1977, which is the year that I was born, and that I get to use that same tool, even though it's been changed a lot, but it's still Alvin and it's still the same vehicle, 40 years later. It's really cool, and you feel like you're a part of the whole big story.
Researcher Jason Sylvan teamed up with colleagues in 2019 on a series of Alvin dives, exploring how microbial communities near hydrothermal vents change over time, specifically when these vents stop venting.
Populations you see on active vents look very different from those on inactive vents. But when you collect a vent that's already dead, you don't know how long it's been dead. And so this experiment involved going to the seafloor, collecting a lot of samples from those vents and putting them into these chambers that we then left on the seafloor for periods of a week and a year so that we knew exactly how long it had been since the venting stopped. And then we bring it back to the lab and we extract all of the DNA from kind of all of the microbes that are living on that rock.
Jason's team then sends this community DNA out to a sequencing facility for analysis.
That tells us not only what microbes are present, so basically the identities of the microbes, how abundant they are, is it Jack and Jane or Jill and Mike or all of them? And so you could really put together the whole story of what a community is doing.
In addition to ongoing research like Jason's, the most exciting discoveries and significant findings may yet be ahead with Alvin's latest upgrades.
There are environments that we're going to be able to go to that we didn't have access to in the past. For example, there's a new type of volcanism that's been discovered in the oceans called petite spots.
These are places in the deep ocean where an oceanic plate dives underneath another one. There are these little structures that pop up that look like small seamounts. They're almost completely unexplored, but now Alvin's going to open up this new door for us to be able to get to these places. We're going to figure out background standard data for what places look like now, because we know the deep ocean is changing. We see climate change having a big impact. There's so many questions we have and so many issues that we have. Alvin's going to be a primary tool to meet those challenges.
The plan is for Atlantis, the mothership, to complete its midlife refit and refresh in July of 2021, and we'll see it come back into service with the certification dives, follow that with some science verification where the community will use the sub, test out its new systems, and then we'll go right into the routine science operations that the sub does.
With Alvin now poised to continue its track record of groundbreaking research, many who have been touched by its scientific discoveries point to an even bigger revelation.
Alvin has brought together geologists, geochemists, microbiologists, macrofaunal biologists, virologists. So many ologists have come together because of Alvin, wanting to use Alvin together, understanding the system. And so the mothership Atlantis becomes a microcosm of the scientific community. This is astounding. This vehicle has done this over time for us and developed relationships and collaborations that led to even greater discoveries. And so I always give Alvin credit for not just discovering vents, but taking the human ideas of science and bringing them together with other people and pushing science forward, really accelerating the pace of discovery.
We are very fortunate to be able to have these experiences. We really want others to have that opportunity.
This is a tool for the entire research community. Anyone with a good question should be able to get into Alvin and try and answer it.
As Alvin continues to mature into the world treasure that it has always been, maybe there will be more opportunities for people of different walks of life to experience the ocean in Alvin.
It's a small community, but it's a growing community, and I'm really looking forward to having other people have that experience with diving in Alvin.
Florida. It's an angler's paradise.
Florida likes to think that we are the sport fish capital of the world.
The value of recreational fisheries in Florida overall is around $12 billion a year.
Tarpon and snook are two of the prized game fish the Sunshine State is famous for.
There's a couple reasons that tarpon are so enticing. One is their massive size. They can grow upwards to 250 pounds, almost over 7 feet long.
It's a fish that migrates great distances. They live for a really long time.
He's big, he jumps. It's kind of like almost the perfect fish.
The adrenaline rush of it just really makes it super exciting and a lot of fun.
The great thing about snook is that they're ambush predators. So no matter what you're using to fish, you're gonna see a show.
You got him, yes.
It's so much fun to catch a snook. You'll see the bite, and then they'll be airborne, just like a tarpon.
Without them, Florida wouldn't be the same.
When they're young, both tarpon and snook rely on murky, mangrove-lined tidal creeks and backcountry ponds.
I would call them essential fish habitat.
Clearly, as more and more people come here and it gets more and more developed, these kind of areas are going away.
Mangrove forests are inhospitable to humans, and with nearly 1,000 people moving to Florida every day, many have given way to coastal development.
The biggest threats to coastal fisheries are habitat loss and degradation.
Even with snook and tarpon being mostly catch and release, we're still seeing declines. What that tells us is it's much more than people fishing.
Habitat is absolutely crucial.
How do scientists study the environment juvenile snook and tarpon need to thrive? And can habitat restoration help ensure the future of the fisheries? In Charlotte Harbor, on Florida's southwest coast, tarpon is king.
Charlotte Harbor is home to Boca Grande, which is the world-renowned tarpon capital of the world.
We have so many fish. In 2010, there was an economic study that found that just locals fishing for tarpon, so not including the guides or people coming in from other states, equated to $110 million annually that was put right into this community.
And it's not just Boca Grande Pass that attracts anglers to this area. Miles of shallow mangrove creeks and flats are a favorite with fishermen as well.
It's just a phenomenal place. If you close your eyes for 30 seconds, you could be completely lost if you don't know where you're going. And that to me is what I really love, is getting in the backcountry like this and just being in that raw Florida.
Slow it down, distribute fast, 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 fast. You got him, yes, that's a snook.
Since 1989, biologists with the Florida Fish and Wildlife Conservation Commission, or FWC Charlotte Harbor Lab, have tracked the abundance of recreational sport fishes and their prey. This fisheries independent monitoring program consists of year-round monthly sampling in Charlotte Harbor.
We are a long-term monitoring program. Our sampling protocols stay the same so that we can track trends in populations over time. In Charlotte Harbor, every month we're pulling roughly 120 nets.
Since 2015, the scientists are also setting seine nets in tidal creeks that are important nurseries for juvenile snook and tarpon.
We set the seine net in a semicircle along the shoreline with one person at one end and then the other person at the other end. They will walk along the shore until they meet, and then they will pull that net in, and we will see what we've caught. A dozen?
Or more.
Or more tarpon?
All young of the year tarpon. We have a snook here. This fish is probably about a year old. We will measure in standard length, which is to the end of the vertical column right here. So this fish is 306 millimeter standard length. Fork length is 352, and the total length is 387.
We will identify all of the sport fish and select invertebrates, and then we will measure most of them and we release them. Our sites are randomly selected. We're not going out to spots where fishermen know that there's snook or know that there's no snook. We are trying to cover the entire estuary. And then we're stratifying by habitat type to make sure that we're sampling in a variety of habitats that these sport fish are using.
This includes remote mangrove-lined ponds, which are difficult to reach.
We'll get measurements on all these.
We caught 11 tarpon and 6 mine cichlids. All the tarpon that we caught were all young of the year fish.
What we'll do at each site is we'll record the type of mangrove and how inundated those mangroves are at the time of sampling.
The juvenile fish find their way into these backcountry waters in the summertime, after the adult tarpon spawn offshore and the adult snook spawn along beaches and the inlets to the estuaries.
That's when we have our rainy season, and that's when we have our highest tides. So the larvae will ride those tides up into the tidal creeks, and then they will swim to the headwaters of the tidal creeks, and they will swim around the mangrove roots to get into these back ponds, pretty much as far back into the landscape as they can go. There's a vast mangrove forest that separates these ponds from the creek, so there's just a small amount of water that connects these coastal ponds to the tidal creeks. They're looking for shallow habitat with protection from predators and an abundance of prey fish. They also, when they're juveniles, can tolerate low oxygen water.
Some of these remote ponds are completely cut off from the estuary during the dry season, giving the juvenile fish a chance to outgrow predators that await them once access to the creeks is restored. Another group studying tarpon and snook in the backcountry is the Bonefish and Tarpon Trust.
It's estimated that we've lost about 50% of our mangrove habitat in Florida. So when you put it in terms of juvenile habitat, you've lost about 50% of your juvenile habitat.
To the extent that overall population of tarpon and snook is dependent on the amount of juvenile habitat, we'll never, ever have as big a population of those species as occurred, say, 100 years ago. We've been spending decades managing the fishermen, and we've been losing habitat, and that's why the fisheries are declining.
You could take every human out of here, take every boat out of here. If you lost the habitat, you've lost the fishery.
One of the things that a lot of people don't understand about fisheries management is that it's entirely about how many fish are harvested, what their growth rates might be, how many are left in the water. There's a real recognition of the need for habitat management as part of fisheries management. It just hasn't been implemented. And we're at the point where if we don't implement it, the game's over.
There are many steps that would need to be taken to make the leap to ecosystem-based management, and we're currently at the beginning phases of that. We are collecting the fish data.
Got a snook at 67.
We're also collecting data on the types of habitats that they are using.
Bottom type is mud.
We are relaying that information to the fisheries managers.
We think that working with FWC on snook and tarpon, because we know a lot about their habitat use, will teach us how to do that. So as new development happens or as different zoning happens, it can incorporate the importance of the fisheries.
In the meantime, the scientists are focusing on restoring impaired habitats. One such project focuses on an area known as Coral Creek on the Cape Hayes Peninsula, just south of the community of Rotunda.
This particular location was developed as residential community with saltwater access, which is why the footprint looks like a canal system that you would see in any other coastal community. The development project started in the 1960s and ran out of funding, so the canals were left abandoned. Currently, the state of Florida owns the land, and it's managed by our Florida Department of Environmental Protection, or DEP.
The initial restoration plan was to fill in the canals and return the topography to pine flatwoods. But then, a group of state employees discovered tarpon rolling in one of the abandoned canals. Knowing this, the Southwest Florida Water Management District, which is the agency tasked with carrying out the restoration, encouraged the biologists to come up with a new restoration design that would be beneficial for the fish.
We brought the biologists together with the engineers. Everybody realized that this was a really unique opportunity. We could set the different canals into an experimental design to test what characteristics of the habitats were better for juvenile snook and juvenile tarpon.
Because we had 6 different canals to work with, we decided to go with 3 different design treatments and duplicate each of those. So 2 of the 3 designs have what we are referring to as a sill mouth. This means that the mouths of the canal are built up higher than the elevation of the rest of the canal, so that water is only flowing through during higher tide events or during storm surge events. What this does is prevents larger predators coming into these nursery habitats when the juveniles are coming in as larvae.
We also wanted to look at depth of the water body. Snook and tarpon tend to utilize slightly different sections of the ponds. The tarpon tend to hang out in the deeper holes, and the snook tend to hang out more at the edges of the marsh.
So in one of the sill mouth treatments, we included a deep hole right after the sill with a shallow meandering creek behind it. Now, the reason that we have the deep hole is because tarpon, as the water gets colder on the surface, they can find warmer water down at the bottom of the hole, and vice versa. As it gets hot in the summer, they can cool off in the deep hole. To test that deep hole theory, we created the second treatment to have the sill mouth, but without the deep hole. So it's shallow throughout with the meandering creek system on the tail end. The third and final treatment lacks that sill mouth, so the water is open and flowing year-round. It also does have that deep hole, again with the meandering creek system.
To see how well the restoration is working, the Bonefish and Tarpon Trust scientists capture and tag tarpon and snook in all of the canals each month.
Each snook and tarpon that we capture, we scan to see if it previously has a tag implanted. This one doesn't, so we then measure the standard length, 251, which is to the end of the vertebral column. Then we measure fork length. This guy is 280 millimeters. Since this guy doesn't have a tag, we'll go ahead and put one in. First, we pull a couple scales, make an incision, pop the tag right into the abdominal cavity, and then let him go.
These pit tags work just like the microchips used in pets. Each tag has a unique ID number, which the scientists record. By tagging the fish over a period of 2 years, the experts can determine how many tarpon and snook are present, if they survive, and how much they grow.
So if we recapture a fish, we're able to see how much it's grown from the time that we tagged it to the time to recapture. Growth is arguably one of the most important characteristics for a juvenile fish. As they get bigger, the more things they have available to eat, and the fewer things are able to eat them.
Past research has shown fish grow faster when they have access to a diverse diet.
We do lavage, or as we like to call it in the field, puke them, pump their stomachs. We can look at what they're eating compared to what's in the creeks and see if they have a particular preference. Because what we've found in the past is that altered habitats have fewer prey available, and the snook in the natural habitats are eating twice as diverse a diet. So right here we have a sailfin molly, but there are also some larger scales here. We take all this back to the lab. We can look at the scales and figure out what type of fish it came from, and we can use that to recreate the diet.
The scientists also use the pit tags to track the movements of the fish. Inside the entrance of each canal is an underwater antenna array that can detect the implanted pit tags and identify fish as they swim by.
Think of it as an automatic toll booth system.
These antenna arrays, they track the fish's movement in one canal, out the other canal. We'll be able to see if it's more productive for them to be secluded or if they need that ability to swim in and out through the open mouths of the canals. We also have an antenna array system that connects to the open estuary, which gives us the final tally on immigration.
While the Bonefish and Tarpon Trust science team is targeting juvenile snook and tarpon in these restored canals, the FWC biologists count and measure all the fish found at the site.
So we caught a couple different species of mohara. We had some striped mohara, some Irish pompano. We caught a Gulf killifish, Gambusia holbrooki. These are all important forage fish for juvenile snook and tarpon.
And we also got a juvenile snook in here as well. He is a new recruit. It's a young of the year juvenile snook.
Prior to the restoration, the Bonefish and Tarpon Trust scientists conducted fish sampling for 16 months at each of the canals.
That way we can compare before and after restoration to see which specific treatments are more successful for nursery habitat.
It's too soon to say if the restoration is successful, but the biologists are already seeing encouraging signs.
From the beginning, juvenile snook and tarpon have recruited to these habitats, and there is an incredible amount of prey fish for them there. We're seeing all of the same forage fish in the restored ponds as we're seeing in the natural habitat.
We're catching a lot more juvenile snook and tarpon than we were before the restoration. So that suggests it's successful. But it's not about the number of juvenile tarpon and snook you see in those creeks. It's how many survive to leave for the next life stage. So if we see a lot of those fish going past the antenna and leaving those backcountry spots to join the adult population, then that's a success. And that'll take a couple years to figure out.
Tarpon and snook also occur on Florida's east coast, where the juveniles encounter a unique set of challenges.
There's very, very little left of a natural ecosystem in the Indian River Lagoon. Compare this to Southwest Florida, where a lot of the habitat is still relatively natural.
The Indian River Lagoon, most of its mangroves, something like 85% are in impoundments.
This entire area before the 1950s was known as mosquito country. It was actually unlivable for most people. Before the impoundments were here, the salt marsh mosquitoes made life very challenging. They would come up in literally the billions.
Starting in the 1950s, berms were built around the salt marshes and mangroves in this part of the state to help control rampant mosquito populations and make the region more habitable for humans.
We have these circular impoundments. When the water levels start going down, they fill them up with water. The salt marsh mosquito needs exposed mud to lay their eggs and to reproduce. And by having water in these impounded areas year-round, it essentially interrupts the life cycle of the salt marsh mosquito.
And it did a really good job of inhibiting mosquitoes from breeding. There are well over 100 mosquito control impoundments on the Indian River Lagoon.
By the 1980s, several scientists and managers realized the impoundments were nurseries for much more than mosquitoes.
There are nurseries for tarpon, for snook, for all different kinds of fish species, and isolating all of these nurseries was not good for the fish populations.
So they came up with a project of putting culverts in and connecting these impoundments back to the lagoon system.
This made it possible for fish to move back and forth in the wintertime when water levels are high.
Then in the summertime, they'd close off the gates, pump the impoundment full of water, and now they could control mosquitoes. And they just assumed that as long as they had the impoundments open for much of the year, they served perfectly good nursery function. Well, about 5 years ago, we decided to really test that and see when and how do juvenile fish leave these impoundments and go back and join their regular population.
To do so, the scientists teamed up with the Indian River Land Trust, which owns many of the mangrove shorelines where mosquito impoundments can be found.
To date, the Land Trust has protected 1,200 acres and 12 miles of lagoon shoreline. Probably about 700 or 800 acres is in mosquito control impoundments, and each impoundment is managed by the Indian River Mosquito Control District. They're a great partner because they understand that science should always be looked at so that we can try to manage these impoundments the best way we can for the fish and wildlife habitat.
Past research in unaltered mangrove creeks showed that snook tended to leave their juvenile habitat after a year.
Which is the next summer after those larvae came into the creeks. They didn't leave the creeks during the winter. So that means that the impoundments are disconnected from the estuary when the juveniles need to come and go.
Knowing this, the scientists set up an experiment at the Indian River Land Trust's Bee Gum Point Preserve.
The basic idea was to, let's catch juvenile fish, let's put little tags in them, and let's track their movement in and out of these impoundments. And much to our surprise, even though we tagged hundreds and hundreds of juvenile fish, very few of these fish left the impoundments during the fall, winter, early spring when the impoundments were open. But shortly after they closed off the impoundments, pumped them full of water, now we're getting all kinds of detections on our antennas on the inside of the culvert saying, hey, I'm looking for a way out of here. And they couldn't get out, they were stuck. So we were perplexed at first what to do. And we talked to mosquito control and they agreed to open up the impoundment, open up the gates, allow water to go flooding through for a week, and then they'd close the gates off again, pump it full of water and go back to controlling mosquitoes. And it was amazing. Within the first 24 hours of opening these floodgates, we had massive numbers of juvenile snook, juvenile tarpon, all flooding out of the impoundment. Once the juveniles reach a certain size after about a year, they can no longer grow well in this mosquito control impoundment. So it's time to move on.
Given the success of the experiment, the research is now being expanded to other areas.
We're repeating it in Bee Gum. We're going to do it in 2 other mosquito control impoundments here in Indian River County. And if it's effective in all 3 impoundments, now we've got something we can go to the managers and say, hey, look, this is really worth adding to your management strategy playbook.
And the Mosquito Control District is willing to work with the Land Trust to open these culverts periodically in the summer to allow for the fish immigration.
The Sunshine State's unique coastal ecosystems and the fishing opportunities they provide are unrivaled in the continental United States.
One of the things that brings a huge number of people to Florida is the quality of life.
Certainly you come to Florida for the weather and for the water.
But as more and more people head south to experience the Florida lifestyle, many of the natural qualities that attracted them in the first place are in decline.
That lifestyle is entirely dependent on a functioning ecosystem. So as those habitats are degraded, our quality of life declines as well.
We want to be able to protect it and keep it going for generations.